Hi all, Dr. Clark here again. Today's general biology lecture, we're going to talk about the electron transport chain. So if you remember from previous lectures, we talked about glycolysis, so the breaking down of glucose molecules without the presence of oxygen. And then we talked about the processing of a product of glycolysis, which is called pyruvate, and pyruvate dehydrogenase. And then the product of that being acetyl-CoA, and the processing of acetyl-CoA in the citric acid cycle, or the Krebs cycle. And now we're going to talk about how the intermediate molecules, or the electron transport molecules, make it through the electron transport chain, and the energy that's utilized by that movement of electrons and protons to drive the production of ATP. So if you remember, glycolysis happens outside of the mitochondria in the cytoplasm or the cytosol, and if it's a bacteria, an archaea, there is no mitochondria, so it happens in the cytoplasm, and will generate a little bit of ATP and pyruvate. That pyruvate then enters the mitochondria and can be processed in multiple ways and eventually makes it into the citric acid cycle or the Krebs cycle in the form of acetyl-CoA. Throughout that cycle, the carbons there's 2 carbons on acetyl-CoA, that they get removed as carbon dioxide, which is not shown here, but that gets removed through respiration of organisms, and the energy from that gets converted into NADH and FADH2, which is also not shown on this. Now, those electron carriers can then make it to the electron transport system, in which protons are going to be pumped across the gradient, shown here, and then re-enter the gradient through ATP synthase, shown here. Okay, so that's just kind of an overview. We'll look at this in a lot more detail in this lecture. Alright, so again, remember when we were talking about glycolysis and the Krebs cycle and dehydrogenase, we said that there are electron molecules or electron carriers that were produced during those processes. The 2 that are produced is NADH and FADH2. Okay. Now, those electron carriers will move electrons and protons, that hydrogen ion, to the electron transport chain. In doing so, the electrons can be used to pump protons across in this case, across membranes inside the mitochondria. The proton pump that we're talking about in cellular respiration is very similar to the proton pump we talked about in photosystem 2. So remember that we had electrons in photosystem 2 that were moving through the protein. When it moves through the protein, it pumps hydrogen ions from one area to another to develop a high concentration of hydrogen ions on one side of the membrane. Now, when these electrons move, in the case of cellular respiration, when the electrons move through the electron transport chain, those electrons have to go somewhere. And when they make it to the last protein in the electron transport chain, the electrons are donated to oxygen, and the protons that are being pumped back across the membrane, and we'll look at this in a second, are going to bind to that oxygen with the electrons to form water. We call this metabolic water, because this water is a byproduct of cellular respiration. Now, for this entire process to work, remember, this is oxygenated cellular respiration. You have to have oxygen to accept those electrons that are free and to pick up the protons that are coming back in to the mitochondrial space. Okay? And so we often call this oxidative respiration. And we'll talk about ways at which, we already did talk about ways at which organisms can do this without oxygen. But most organisms that are highly functional and highly complex will go through oxidative respiration. So they're taking in oxygen. This includes, plants that, I told you that oxygen is a waste product, but they can utilize that waste product to drive cellular respiration. Plants are still doing cellular respiration. They still have mitochondria, and they still need the oxygen to drive that process. It's just they produce more oxygen than they could ever use, and that's why they get rid of it through their stomata. Right? And it is poisonous to them unless they can bind it up to form metabolic water. And we'll look at some examples of this in a second. So here's just kind of the general overview. This is going to happen in the mitochondrial space or mitochondrial membrane, between the mitochondrial matrix and the intermembrane space. So the mitochondria has two membranes. We've talked about this before. Organelles, a lot of organelles, have double membranes. And so this is happening between the mitochondrial matrix, the inside of the mitochondria, and the intermembrane space. NADH is going to donate electrons to a protein complex. So these are transmembrane proteins that are going through the lipid bilayer. As the electron moves through the protein, protons are pumped across this gradient to make a high proton gradient in the intermembrane space. FADH2, which is a different electron carrier molecule, is going to donate electrons also, and we call this protein complex 1 and protein complex 2 because these different intermediate molecules are donating electrons at different areas. But the electrons eventually get carried to protein complex 3, where the electron leaves, goes back into the mitochondrial matrix, binds with oxygen and the protons that are being pumped back across, to form metabolic water. Okay, so this is what we call chemiosmosis, okay, or osmosis, which is driving phosphorylation. So remember, osmosis is just movement of material from a high concentration to a low concentration. So in this case, we're using electrons to make that high concentration and then allowing for them, the protons, to be brought back across the membrane through one protein called ATP synthase. So again, NADH, FADH2, that energy that is being removed from them by taking the hydrogen, the protons away, and removing the electrons is what's driving these proton pumps. It's pushing the protons across that membrane. And like I said before, those protons, they're in that intermembrane space, and they're in high concentration, and through osmosis they need to move, or they want per se to move back across that membrane. There's too many protons in one area, so they gotta move back across, and the only way they can move back across, at least the only way that we've discovered that they can move back across, is through ATP synthase. That enzyme that drives ATP production via chemoosmosis or chemiosmosis. Okay. So here's the whole system. So again, this isn't showing glycolysis because this is oxidative respiration. But remember, glycolysis would happen in the cytoplasm to form pyruvate. And you're going to get a little ATP from that. Remember, you get 2 ATP molecules from the formation of pyruvate, 2 molecules of pyruvate. Pyruvate then would enter the mitochondria. When it enters the mitochondria, it's going to be processed and goes through pyruvate dehydrogenase to form NADH and acetyl-CoA. Remember, if there's not enough energy in the system, acetyl-CoA will not enter the Krebs cycle or the citric acid cycle. Instead, acetyl-CoA will go into fat synthesis mode. And so the carbons from this will be donated to a hydrocarbon chain to form a fatty acid and basically form fat storage. That acetyl-CoA, if there's not enough energies, would enter the Krebs cycle, go through all the changes that we talked about in the Krebs cycle. Okay, so bind to that 4-carbon molecule to form a 6-carbon molecule, and then one of the carbons is lost or is removed for carbon dioxide, NADH is formed, another carbon is removed for carbon dioxide, FADH2 is formed, a little bit of ATP is formed through the movement of the molecules and the breaking of the bonds is enough to phosphorylate ADP. But then these electron carriers, this is the energy portion of our food. This is where we get most the bang for our buck. And that comes in these electron carriers, which then take those electrons to the electron transport chain, allowing for protons to be pumped across into the intramembrane space. And then as this gradient fills with protons, they have to get back across. And they can come back across through ATP synthase. And that allows for 34 ATP to be formed from one glucose molecule. Okay, so one glucose molecule, when it's all said and done, is actually going to form 38 ATP. 2 from glycolysis, 2 from the Krebs cycle, and 34 from the electron transport chain. So if you remember back, I said, in order to create or in order to form one glucose molecule in photosynthesis, we need to expend roughly 54 to 60 molecules of ATP. We consume that 1 molecule of glucose, and we can get about 38 molecules of ATP back. So it's better than 2. And so the benefits of having oxygen in the atmosphere and oxygen driving the system is much better than only forming 2, which is what organisms do without oxygen. It's still not a 1-to-1 ratio. So without the energy from the sun, the free energy, the photons from the sun, the system would be broken. If you had to just utilize carbon-based energy, so coal or oil or natural gas, to run the entire system, there wouldn't be enough energy to do that. Right? You would be at a net loss. So instead, we're at a net gain because the photons that are coming in, they're free. That's free energy. We don't have to pay for that. Right? In the case of if you're using a power bill or something like that, you do have to pay for that energy, and you would be at a net loss. So at any rate, the other piece of this is that electron, or those electrons, have to go somewhere. Right? And they do. They get pumped through another proton or another protein and get donated to oxygen and the protons from the pumping of the protons through ATP synthase to form metabolic water. And most of you remember when we were doing like bacteria labs, I told you that the bacteria dishes have to be stored upside down. So the nutrient auger has to be stored up. And that's really because when bacteria, which can still do this, and I'll show you in just a second. Remember, bacteria don't have a mitochondria, but bacteria still can go through the electron transport chain. I'll show you how that's done. They're going to produce the same thing as what we would, metabolic water. And if you have it stored upside down, so the gel down, they're going to produce water, and that water will drown out the bacteria, because it has no place to go because it's in a plastic container. So instead, we store them upside down. They produce water. The water lands on the lid. Then you can just pour the water off the lid, and there you have it. Another reason, or another piece of evidence suggesting that bacteria can do oxygenated cellular respiration. And I'll show you that in a second.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H+ and FADH2. In the mitochondria, the electrons from NADH plus H+ are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Okay, so real quick, I just want to kind of go off a little bit of topic, and that's really talking about, this molecule, well, this kind of whole complex, the cytochrome C or cytochrome B complex, or really cytochrome anything complex. When we talk about evolution, we'll come back and talk about these because these transport systems occur in almost every single organism on the planet. Same proteins, same molecules, which allows for evolutionary biologists to look at if all eukaryotes have this system, then when was the last time, say, a human and a kangaroo shared the exact same ancestor or shared the exact same cytochrome C complex? And so they can look at how many changes has there been in the cytochrome C complex between those two organisms. And if those two organisms haven't been, connected for 150 million years, right? Now remind you that humans have not been around for 150 million years, neither have kangaroos, but that's the last time our ancestors would have shared that cytochrome C complex. This points to evidence that suggests, well, if that's true, then we should also be able to match that to the geological record. So if Australia, where kangaroos evolved, and Africa, where humans evolved, if they're last connected roughly 150 million years ago, then that suggests, well, hey, we got geological evidence and we have genetic evidence from one protein that suggests, hey, these two are comparable. These two are saying the exact same thing, that about 150 million years ago, the continents broke, there's no genetic connection between those organisms any longer, and you got kangaroos over here, and you eventually get humans over here, and now, from that breakage, the cytochrome C complex has changed in a certain number of times. Okay, we'll come back and check that out, but I just want to make sure that you remember that when we're talking about things like molecular clocking and protein analysis, we're often talking about proteins that all organisms have. Every single eukaryote can go through this process and does go through this process. Even prokaryotes have a lot of these same proteins. They're not embedded in the mitochondrial space. They're embedded in their membranes. Okay? So their plasma membrane that surrounds the entire prokaryote still has these proteins that are embedded in it, so they can still go through this process. And that's what we're going to see next.
During glycolysis and the tricarboxylic acid cycle, oxidation of organic molecules results in production of reduced coenzymes such as NADH. These coenzymes transfer hydrogens to the electron transport chain, which is located in the bacterial cell membrane. A hydrogen consists of a proton and an electron. The electron transport chain consists of a series of special electron carrier proteins that shuttle electrons from NADH to a terminal electron acceptor, such as oxygen. Electrons enter the electron transport chain when NADH transfers its protons plus electrons to a membrane-embedded carrier protein. The electrons are sequentially carried along the electron transport chain while the protons are shuttled to the outside of the membrane. Some of the electron carriers, such as coenzyme Q, accept a proton from the inside of the cell membrane as it accepts electrons. The proton is then transported through the membrane as electrons move down the chain. This increases the proton gradient across the membrane and enhances the proton motive force. During aerobic respiration, the last carrier protein transfers a pair of electrons to oxygen at the end of the electron transport chain, and water is formed. The enzyme ATP synthase utilizes the energy of the proton motive force to synthesize ATP. This enzyme allows protons to pass back into the cell and couples the energy released in this process to the phosphorylation of ADP to form ATP.
Okay, so there you can see that bacteria still have the same capabilities as eukaryotes, so prokaryotes still have these capabilities. When oxygen is present, they have the capability of forming metabolic water and doing the electron transport system to generate more ATP, a lot more ATP. And it just shows you that things like ATP synthase, that enzyme, if it's found in archaea, if it's found in bacteria, it's found in protista, fungi, plantae, and animalia, it's ancient. And it gives us another piece of evidence that all organisms came from pre-existing organisms, and they evolved, and some systems just work. Some systems have not changed in a long time because they're efficient enough that organisms can get energy from it. Now, could you design a better system? Of course. If you're expending 54 molecules of ATP, wouldn't you want to get at least 54 from cellular respiration? Of course. But that's not the case. We don't get that. So there's not a total net gain. It's a net loss as if we're paying for the 54 that we start with. But we're not. Okay? And that's from the sun. All right, so remember what I said before, that we're not just talking about glucose, okay? We're not just talking about carbohydrates. We're talking about the 4 biomolecules: proteins, lipids, nucleic acids, and carbohydrates. Those are your main 4 food resources. And so how do we get energy from all of them? Well, it depends on the molecule, and it depends on what subunit you break that molecule down. So again, most of us are not consuming glucose directly. Most of us, humans at least, we're consuming carbohydrates in different forms: starches, sucrase, lactose, other forms of glucose. Now, a lot of those have to be converted into glucose molecules or into other intermediates that are similar to glucose. So, we talked about, sucrose before. Table sugar has to encounter sucrase, that enzyme broken down into fructose and glucose. Now, the glucose molecule can enter right into glycolysis, but the fructose molecule has to be converted even further. It has to change just a little bit to go into glycolysis. The same thing goes for proteins. You're consuming proteins. You eat that protein, okay? It doesn't just enter into energy. It has to be broken down into, that tertiary structure or quaternary structure has to be broken down into simpler structures, amino acid chains. Then you have to break the peptide bonds. Then you have to break up the amino acids. You have to remove the amino groups, and then they can enter the Krebs cycle. And at that point, then you can start getting energy from protein. Same thing with lipids and other things. They have to enter as simpler subunits. Each subunit can enter the cellular respiration cycle at different places. And so here's just kind of an overview of where those would enter. So nucleic acids, every time you eat an organism that was alive, you're going to take in some nucleic acids. You're going to eat their DNA. Okay? And so that would be converted into nucleotides. Those nucleotides could enter the Krebs cycle directly, or they could just be a metabolic product, meaning often a metabolic waste product. Okay? Protein, same deal. Going to have to be broken down into amino acids. You're going to have to strip or deamination. You're going to have to strip the amine group from it. Some amino acids can enter as pyruvate. Some amino acids are going to enter the Krebs cycle directly. Some are going to be waste products. Okay, polysaccharides, same deal. Going to have to go through glycolysis, pyruvate, acetyl-CoA into the Krebs cycle. Lipids, same deal. Fatty acids, you have to oxidize those fatty acids, acetyl-CoA, and then into the Krebs cycle. So no matter what energy source you're going to consume, pretty much, if you're consuming a large amount of lipids or a large amount of proteins, they're going to enter as acetyl-CoA. And eventually, if you have too much energy, no matter what, it's going to be stored as fat. Doesn't matter if you're consuming too much protein, too much lipids, or too much polysaccharides. Okay, so removing certain groups from your diet is never a good thing. Now we're going to look at a little video about individuals who overconsume protein. This is often the case of people who lift weights a lot, bodybuilders, sometimes pro athletes, wrestlers, individuals that buy into the protein shake, protein bars, protein everything. They're probably doing a lot more damage to their body than good to their body.
Dietary protein and amino acid intake must meet the essential amino acid needs of the individual. But any intake in excess of about 1 gram of complete protein per kilogram of body weight cannot be incorporated into protein by the body. Since the body has no means of storing protein or amino acids, any excess is converted to glucose or fat and used or stored as such. The conversion of amino acids to glucose or fat requires that the amino group be stripped from the amino acids. This nitrogen must then be eliminated, putting additional stress on the kidneys. For a 180-pound male, consuming a diet made up of 55% carbohydrate, 30% fat, and 15% protein, only about half of the ingested protein, 8%, is used to build muscle. The rest is used for energy. Adding more protein to the diet, now up to 20%, does not change the amount used for building muscle, but the amount used for energy increases. This is even more evident as we change to 25% and 30% protein in the diet. The same trends are apparent for a 140-pound female. Note that the amount of protein used to build muscle stays constant, but the amount used for energy increases as we increase the dietary intake of protein from 15% to 30%. Since increasing protein intake cannot build muscle and does add a burden to the kidneys, most nutritionists advise people not to eat more than 2 times their calculated RDA for complete protein.
Okay, so that brings me to a point. So, they're talking about 1 gram per 1 kilogram of body mass. So what this amounts to is if you're taking like a 180-pound male, that individual can consume roughly 80 grams of protein, and that will be converted to muscle. What that amounts to is about 8 or so ounces of chicken breast. Okay, so 2 4-ounce chicken breasts, that's about your 80 grams of protein right there. If you're consuming more than that, all you're going to do is take that protein and convert it into glucose, and then utilize that as energy for your muscles. But in doing that, again, you're putting strain on your kidneys. So for those individuals out there with protein shakes and going to call me a liar that they're building muscle with protein shakes, okay, this is probably true. You are building muscle, but it's not the protein shake that's doing it. It's the fact that you're lifting weights and you're burning up that glucose that you're converting that protein into. So for an experiment, for those of you who think I'm lying, try this. Instead of doing the protein shake, instead of doing the excess protein, eat your 80 grams if you're 180, or calculate it out. If you're 200, you're going to eat, closer to 100 grams of protein. But calculate that out, do the minimum that you can convert into muscle, and then instead of doing more protein, do more carbohydrates. Just eat sugar. And I'd be willing to bet the muscle production is identical. This experiment has been done by lots of people. Okay? And if the muscle production is identical, then all you're doing is you're saving your kidneys the stress from breaking down that extra nitrogen. All right, with that, next time we are going to talk about mitosis and meiosis and how cells generate or regenerate themselves. All right, so next time.